Hi guys, thanks for joining. I'll start the class in just 2 minutes once a few more people have joined. Just hold on till then. Okay, I think we can get started with the class. I'll just share my screen. Right. I hope that's visible to everyone. I think you can see it. Anyhow, okay. Uh, so welcome everyone to today's problem solving session. We'll be tackling. So this is the fourth week, and we're looking at the fifth week of um, course material that was uploaded, and we'll be solving some questions in typically the MCQ format. Though we do have some questions that are going to be a little more sort of you have to think a bit more for them. So we'll go through them as well, along with some theory, just to revise and remember whatever you've seen in your coursework so far. So let's get started. So this week you guys covered lymphatic system as well as the kidney and renal nephron architecture, as well as how filtration in the kidney takes place, right? So these are some of the important topics, and I'll move through them with you guys. Not everything, but whatever is possible. Right, so a quick recap, because now we're not going to be coming back to muscles and cardiac muscles or skeletal muscles anymore for the time being until we discuss smooth muscles later. So we learned about, so far we've learned about skeletal muscle anatomy, right? We learned about um, the different components of when you go from the macro to micro scale in skeletal muscles. And we learned about the importance of um, the sarcomeres in the skeletal muscles containing your ZH discs and zones, and the anisotropic and isotropic bands, and how this sarcomere is the contractile unit of skeletal muscles and cardiac muscles as well, right? So then we learned about cardiac muscle anatomy, right, where we looked at the slight differences between cardiac muscles and skeletal muscles, particularly with regard to developed T tubules, less developed sarcoplasmic reticulum, as well as branched macroarchitecture of these muscles. So they're not like skeletal muscles, there are branches here. Enhance the electrical connectivity within these cells that allows the heart to beat um, and maintain its rhythmicity by conducting electrical charge across the various heart tissues, right? So then, of course, we talked about heart rhythmicity and electrical activity. Just remove this. Okay, right. And we learned about how the different events during electrical activity in the heart take place. So if you're sitting at the ventricles, the ventricular musculature of the heart, then you see these different phases of depolarization and repolarization. And we looked at the different kinds of channels that open to allow these spikes that move away from the resting voltage, resting potential, and allow these different phases to take place that essentially cause this spike and relaxation in the heart. So essentially, this allows for heart muscle contraction and subsequent relaxation during repolarization phases. And we looked at the electrocardiogram wave very famously, right? The PQRST wave that's there. And we learned about what each of these different regions mean, right? And finally, we looked at hemodynamics, and we looked at how blood pressure and osmolarity is maintained. We learned about nitric oxide, which is a very important modulator of muscle relaxation in smooth muscles that surround blood vessels. So you have endothelial cells that produce nitric oxide, which then diffuses through them because it's a gas, and it reaches the smooth muscle cells and allows for relaxation, which causes dilation of the blood vessels. So more blood will reach that particular tissue. So the body will decide based on the requirement where this kind of dilation mechanism is required, and then there will be local increase in blood supply in that part. We also learned about the renin-angiotensin-aldosterone system, which is also very important for locally changing the blood pressure, and it works via the kidney producing renin, then the lungs producing the ACE enzyme, right? And then aldosterone being produced from the uh, this, just a sec. Yeah. So yeah, so we learned about the RAS system and how this how ACE inhibitors work to lower the BP by inhibiting ACE enzyme, right? So we learned all of these different things, and now we're going to leave all of it behind and move into the lymphatic system. So if anyone has any questions regarding any of the stuff that's been covered previously in the last couple of weeks worth of lectures, then you can, of course, ask me now, or you can ask me in the messages and stuff. Okay? All right. Oh, yeah, here it is. um, cool. All right. So we'll move to the main topics. So the lymphatic system, right? So what is the lymphatic system? So you've all heard about the lymphatic system. It's another of a major arm of the circulatory system, except this is not blood that's circulating, it's lymph fluid, right? Which is essentially this tissue fluid that filters out of the blood and then enters into the lymph vessels and then is circulated. And it contains a lot of fat, and it's very important for the immune system because you have a lot of lymph filtering through lymph nodes, which picks up a lot of antigens and allows immune cells like T cells and B cells to experience antigens at what we call as immunologically relevant sites. So actually, I work in immunology. So the extra class that we take typically as a part of the course, I plan to take in immunology. So if anyone's interested, you can join it as and when it is announced. Right. So in this image here, you can see that there are these blind capillaries. Blind just means that they're just ending here, right? They're ending in closing here. So they're not opening out in tissue like that. They're closed. So it's like a dead end kind of. So these blind lymph capillaries in green are shown here, and they are all surrounding these capillaries, and these are the blood capillaries, and the blood that's moving through here in these really narrow capillaries is under high pressure, so we have a lot of tissue fluid that flows out of them, that sort of leaks or trickles out of them and gets in between the tissue spaces, in between the tissue cells, and it's called tissue fluid. Okay, and so this tissue fluid then actually moves into the lymph capillaries. So the smallest lymphatic vessels are these lymph capillaries, which begin here in the tissue space, and they're what they call blind-ended sacs. Okay, so the lymph capillaries are found in all regions of the body except for a few places, like you don't see them in the CNS or the central nervous system, and you don't see them in the bone marrow, right? And you also don't see them in certain tissues like in the epidermis, the highest level of your skin, as well as because that also doesn't have blood vessels. Um, so wherever there are a lot of blood vessels, aside from these particular examples, you would see lymph capillaries as well, because there's always going to be tissue fluid accumulating in those regions. So how is it that this tissue fluid is entering into the lymph capillary so easily? So the wall of the lymph capillary is composed of an endothelium, which has these simple squamous cells, and they overlap in a way that forms a simple one-way valve. So what does this mean? Essentially, if the tissue fluid enters the lymph capillary, it shouldn't keep fluxing in and out of the capillary. It should remain inside so that it flows. So just like how in veins you have valves that prevent backflow, Similarly, here you have this kind of an overlapping cell architecture that allows only one-way movement of tissue fluid. So this is what permits fluid entry but doesn't allow it to leave the vessel. And interestingly, what's different between lymph vessels and blood vessels is lymph vessels only carry fluid away from the tissues. That makes sense, right? Blood vessels like arteries, they will carry it to the tissues where the oxygenated blood will diffuse in the tissues. Allow oxygen to diffuse in the tissues, and then the veins will carry it away. So both to and from is done by blood vessels for blood, but lymph vessels only will carry it away. You don't distribute lymph into tissues. Okay. Um, right. So that's about the lymph capillaries in the tissue spaces. So now we're going from the micro level to a macro level. So what does that mean? We have these microscopic lymph capillaries over here. They merge together to form lymphatic vessels. These are larger, okay? So these lymphatic vessels then join further to form larger tributaries. Like rivers have tributaries, these are called tributaries, and they are specifically called lymphatic trunks. So these are even larger. You can see them with your eye. And these drain large regions in the body. So as we know, lymph fluid or tissue fluid is accumulating everywhere because of pressurized filtration. And so you have the requirement that lymph fluid does not accumulate in regions, and it does happen in people who have certain lymphatic drainage issues, have certain disorders. It happens in, a lot of times you might see this in elderly people or patients who have like swelling in their hands and feet. It could be because of lymph fluid accumulation, and that's because there's some problem or the other internally that's leading to improper drainage of lymph, right? So you need all of these mechanisms intact for proper drainage of lymph from the tissue, or you will have edema or swelling. So yeah, so the lymphatic trunks, these drain like large regions, and they merge into the lymph until the lymph enters the lymphatic duct. So there are two major lymphatic ducts in the body. You have the right lymphatic duct and you have the thoracic duct. Okay. So the right lymphatic duct drains lymph from the upper right quadrant, so here, of the body, and the thoracic duct drains all the rest of it. Now, why is it so unequally spaced out? I don't know. You'll have to look it up. That's a question that maybe you could try to think about. Uh, like veins, for instance, the lymphatic tributaries, oh, sure, I mentioned this, that they have very thin walls and valves and all to prevent backflow. Now, the thing is, just even veins, they don't have necessarily a pump like the heart. Like in arteries, you have very strong pumping, so the arteries themselves have heavy musculature that allows this pumping. Veins have lower pressure, but they still have some amount of systolic pressure that allows the movement of blood. But in the lymphatic system, there is no heart like we have in the cardiovascular system. So how does it move forward, right? How does lymph fluid even move? It has to move up, it has to move like in every direction, it has to move anti-gravity. So the body is smart in that way, just like it was mentioned in your coursework. The pressure gradients that move lymph to the vessels come from actually just skeletal muscle action and respiratory movement and contraction of smooth muscle and vessel walls. So no specialized pumping mechanism like in the heart. It's just along with your breathing, you have proper contraction that and skeletal muscle action, meaning basically that if you stand for a really long time or you sit for a really long time, say at like an office job, people just sit with their legs over, like down, and they don't like at their chair and they don't move their legs too much. That could actually cause major problems because that can cause lymph accumulation. So it's always good to have some skeletal muscle action there so it doesn't cause pooling, right? Because otherwise it's relatively static and can lead to edema. So it's always best to have some movement during the day, skeletal muscle action, alongside your respiration and normal contraction that takes place. Right. So I hope that is clear. So now something I thought I'd go into detail because it is quite useful to know, which is about the lymph node. So the lymph nodes, as you know, are extremely important sites for the immune system, and you have a lot of immune activity that takes place in them that allows the immune system to recognize threats to the body, dangers like some foreign protein, some virus, cancer, even. The lymph nodes are very rich sites where T cells and B cells can exchange and receive information about infections or diseases that may be causing a threat to the body. Okay. So the lymph nodes have lymphatic, they're connected, or wired up to the lymphatic drainage as well as to blood, as you can see here, right? And they have a very well-defined structure. They're also sort of kidney shaped, and they have these distinct regions within them, okay? And you have different types of immune cells that are localized within these different regions, and the regions are very cleverly maintained in such a way that there's actually regions where they can interact with each other. So these regions, these follicular regions, they have a lot of B cells, whereas the paracortex and cortex regions, they have a lot of T cells. T cells receive information from B cells. So there are these middle regions where they actually are able to communicate with one another. Okay, so and then receive information. So there's some really nice videos that I could show you in the extra class where you can see these T cells and B cells that are, carrying some fluorescent tag roaming around inside the lymph node and interacting with one another and receiving information from one another. So antigens, as you know, could be anything. It could be something from a virus, it could be from a bacteria, it could be from a mutated cell that's got some abnormal proteins on its surface. And so these antigens that may be foreign or maybe just homegrown, right? They can enter the lymph node through the afferent lymphatics. Afferent just means something that's carrying something into that system. So here's your afferent lymphatic in the cortical region. And then and cells and fluid, they leave through the efferent lymphatic. So that's over here. So once all of this interaction with the fluid is done, these cells move inwards and move out from the efferent lymphatics. So the cells, which in this case I'm talking about lymphocytes like T and B cells, also enter through the node from the blood by migrating across specialized, these are called HEVs or high endothelial venules. So from the blood, you can enter via the HEVs. Um, and within the node, the cells then distribute themselves into distinct zones. So you have these germinal centers that allow for B cell proliferation, and they form within these follicles. And T cells are in the paracortex, right? So this region, and they distribute and then they can come to these sort of in between regions that allow interaction between the B cells and the T cells. And these are some fixed structural elements that allow these cells to then move and get out by the efferent lymphatic vessel. So that's how lymph nodes work, and there are many of them, right? So you can find lymph nodes pretty much all over the body, like especially you have a lot of them concentrated in your armpits, groin regions, along your neck. If you ever get like a cold or an infection and you feel the left side of your neck, you might actually feel some bumps on your underneath your skin, which is like inflamed lymph nodes. Not always, but it might happen. Yeah, you have, yeah, that these sort of go up from up alongside your neck. Yeah, and then you also have lymph nodes in the ventral side near your abdomen. You have them inside in Peyer's patches in your intestine. These are not lymph nodes, but these are sort of immune enriched regions as well. So all of these, they don't all have the same structure like in the lymph node, but they function in a similar way. Even in the spleen, you have specific regions for B cells and T cells, and then they sort of mature and talk to each other there as well. Yeah, so that's about the lymphatic system. So I think I have some questions for you guys now. So um, for those who don't know, um, typically how we tackle the questions is I read out the question that will be on the screen. And if you know the answer, or even if you don't know and you just want to try, that's totally cool. You can send the answer that you think is right in the chat, and then we'll discuss uh, the question in more detail. Yeah. So this is kind of a question about whether you know it or not. It's not really too much thinking. So approximately how much fluid is filtered out of the capillaries into the interstitial space over a 24-hour period. So how much tissue fluid essentially is being generated over 24 hours? That's the question. So if anyone knows the answer, you can send the answer in the chat. Does anyone know? It's okay if you're not sure, like there's no harm in trying. Anyone? I guess not, huh? Okay, maybe I'll tell you the answer for this one then. So the right answer here is A option, which is 20 liters. So that's a lot, right? But yeah, the body is able to filter that through the lymph, um, yeah, through the lymphatic system. Anyway, so all right, let's move to the next question. So, what primarily explains the reabsorption of most filtered fluid back into the capillaries? So, which of these mechanisms would be able to explain that relatively well? Does anyone know? Hmm. Okay. Uh, right. Anyway, so the answer is B. It's the Starling hypothesis. So does anyone like need some explanation as to what the Starling hypothesis is? So it's basically a logic that explains fluid movement, and it explains why fluids need to move in and out of capillaries. So it's like the balance between hydrostatic pressure and oncotic pressure or colloid osmotic pressure. So it's like why why do fluid why does fluid leave capillaries, at one end and return at the other, right? At another. So when fluid moves across a capillary wall, it actually depends upon some different types of pressures, right? So one is capillary hydrostatic pressure, right? So that is pressure that pushes fluid out of the capillary. Okay, so that's capillary hydrostatic pressure. And that's caused by your blood pressure. So higher the blood pressure, more the capillary hydrostatic pressure. So arteries would have highest capillary hydrostatic pressure. Then you have interstitial hydrostatic pressure. This is what pushes fluid into the capillary. So it's quite low relatively because you don't need that much for it to happen. And then you have something called capillary oncotic pressure. So this is what pulls fluid into the capillary, and this is caused by different plasma proteins like albumin. You guys have heard of albumin, right? It's a major component of the blood plasma, right? And proteins cannot actually cross very easily. So it's like if the proteins are there, it's sort of like they're pulling the water back in. So these plasma proteins pull fluid into the capillaries. Then you have interstitial oncotic pressure. So this pulls fluid out of the capillaries. So then there is this equation, a Starling equation, that essentially calculates the net filtration pressure, right? So you, I should have made a slide on this, but essentially you are, if you have a net positive filtration pressure, that means that filtration is taking place and effectively, and fluid is leaving the capillary. If you have negative net filtration pressure, you are having more reabsorption, so fluid is entering the capillary. So this is a balance of negative and positive, and that allows filtration as well as reabsorption. So over here, when we apply it to this question, the Starling hypothesis is explaining the reabsorption of most filtered fluid back into the capillaries, because what goes out must then also be reabsorbed back in. Okay, that's the only way that you can maintain these things. So when does it go wrong? Imagine if you have very increased hydrostatic pressure. What does that mean? Capillary hydrostatic pressure is from the pressure of the blood in your in the vessels. That could be because of blood volume, pumping of the heart, so on. So you're during, say, some heart condition, increased hydrostatic pressure has taken place, you can have edema. If you have plasma proteins that are decreasing, um, just a sec. Yeah. So if you have plasma protein levels that are decreasing, you can have edema, because again, absorption of water is not taking place properly into the capillaries. So you don't have proper drainage. Or if you have increased capillary permeability, which happens in the case of inflammation, or some sort of obstruction in the lymphatics, then you can have fluid accumulating in tissues because of improper extruding of the liquid as well as improper absorption. So in any of these steps, if there is some problem with the pushing and pulling of fluid, then that can lead to edema and a certain level of um, disruption of the Starling forces. So that's sort of your Starling hypothesis. Essentially, it's a balance of forces. Okay, so that's about that. Okay, let's go to the next question. So once interstitial fluid enters lymphatic vessels, it is called what? So for those of you who just joined, you can send the correct answer into the chat box and we can discuss it after that. So whatever you think is the answer to question 3, do send it in the chat. If you're not sure, it's totally okay. There's no harm in getting it wrong. Does anyone want to try? So we were just discussing, right, that interstitial fluid is also tissue fluid. Yeah, exactly. Good. So one person has answered correctly, which is, it is C option, right? Which is lymph fluid. So interstitial fluid or tissue fluid is essentially fluid that filters out of the blood vessels into, sorry, out of the capillaries into the tissue and in between the tissue cells, and then that then unidirectionally enters into blind lymph vessels, right, into the lymphatic vessels. So once it has entered into the lymphatic vessels, exactly, good, it is called lymph or lymph fluid. Okay, so great. Then now that that's clear, let's go to the next question. So which structural feature allows lymphatic capillaries to readily accept interstitial fluid? So we were talking about how this is a unidirectional movement. So do you guys sort of remember the structural conditions of the lymphatic capillaries that makes them kind of unique? Nice. So the answer here is D option, okay, which is overlapping cells with some gaps. So what these overlapping cells with gaps basically mean is that they form these one-way valves. So these gaps allow the fluid to readily move through into the lymphatic capillary, but they also don't prevent backflow. So they don't allow the liquid to move back out of the capillary into the tissue again. So the structure is such that it readily allows it to go in, and that's very important for proper drainage, but it does not let it back out. So it's unidirectional movement. So correct answer here is B. Okay, let's move to the next one. So I just gave the answer away, but why can fluid enter terminal lymphatic vessels but not easily flow back into the interstitial space? So I think now this should be an easy one to get. Yeah, yeah, fair enough. I won't spend too much time on this one. So the correct answer here is A option, which is stretching of endothelial cells causes this flap-like closure, which acts like a valve. So these endothelial cells, they overlap and they form these valves, and they have gaps that allow the fluid from in between. And if there's like a flap, imagine if you're sort of holding your palm straight like a triangle. So it opens in one way, but if the liquid tries to come back, you just flip your hands back down, and that's what the valve is like. Okay, so that's why backflow does not happen. All right, next question. So larger collecting lymphatic vessels propel lymph mainly through which of these mechanisms? Would others like to also give it a go? I had got some answers previously. So the answer here is C. Right. So the answer here is C option. Yeah, so I can understand why you're confused about B because it's like muscle fibers within the vessel wall. But these you have to see the B option. It's written skeletal muscle fibers. And what is the thing with skeletal muscle fibers? They are voluntary. Now it cannot be that your lymph vessels have skeletal muscles, right? We have voluntary muscles that you have control over. Um, Around your lymph vessels, right? That's not possible. So all of the muscles around our arteries and our veins and our lymphatic vessels and our internal organs, these are all smooth muscles that are embedded into the tissue of these organs and blood vessels and so on. They are all smooth muscles. Okay. So there are some low level peristaltic contractions of smooth muscles that allows lymphatic vessels to move through. And if you have skeletal muscle contraction, not in the vessel wall, but in the body, like in your legs, for instance, you have skeletal muscle contractions that sort of keep a certain movement going. Like, for instance, if you're doing cardio or you're walking or you're doing some yoga or you're just doing a little stretch, that kind of skeletal muscle that's not in the vessel wall but that's there in your major skeletal muscles in your limbs and so on, that can help with lymph movement in the lymphatic vessels, so that's there. So I think indirectly cardiac contraction, skeletal muscle fibers in not in the vessel wall but in other places. I think indirectly A and B could be correct here. But the major ones for the larger vessels is the smooth muscles, especially if it's going anti-gravity. Okay, great. So I hope that one is clear now. So we can move to the next question. All right. Just a sec. Ah, okay. So which of the following mechanisms does not contribute to lymph movement? So now we were just talking about it. So maybe think from the perspective of what is required for uh, lymphatic, a proper lymphatic drainage and movement. And I think you will get it easily. Yeah, yeah, I'm glad I'm getting your answers here. So you're absolutely right. The answer is D, which is a dedicated lymphatic heart. Like, what is that, right? There's no such thing as a lymphatic heart. Uh, so we can discuss the other ones as to why they're useful. So we know that respiratory movements, they do modulate lymphatic movement. So there is a sort of synchronization of lymph movement with respiratory movements. Your breathing actually can help move along the lymph in your body. Arterial pulse pressure, that is sort of regulated by your cardiac or your heart contractions, that can then regulate a fluid movement into the capillaries, which can regulate filtration. So essentially, indirectly, arterial pulse pressure can contribute to lymph movement because it can contribute to the pressurized filtration of tissue fluid at the capillary level, and also indirectly um, control how much blood is going to your muscles, for instance, and then that can skeletal muscle contractions can have an effect. And of course, continuous filtration from capillaries is important for lymph movement because um, that decides how much tissue fluid is being produced at the capillary level, right? So A, B, and C, in direct or indirect means, have effects, clear effects, and contribute to lymph movement, whereas D is just a false option. Yeah, cool. Let's go to the next one. So why is an efficient lymphatic system particularly important in the lungs? This was mentioned in your coursework, and I thought it was quite an important point. So you have to remember that the lungs are what is called a highly vascularized organ. What does that mean? Right? That they have a lot of blood vessels. Right? Why do they have a lot of blood vessels? Why do you need a lot of blood vessels? Because of course, lungs are where your gaseous exchange takes place. You need to throw out CO2 and receive oxygen from the alveoli, right? So you need to have, so you have really a high number of blood vessels or vasculature, as they say, in the lungs. Now, as I mentioned, where you have lungs and capillaries, sorry, when you have blood vessels and capillaries, there you will have the lymphatic system, you will have lymph vessels. But if you don't have proper lymphatic drainage in the lungs, what will happen? Right? You will have capillaries that have blood flowing through them and they are at some X amount of pressure. Of course, they're not under as much pressure as they would be, say, in the aorta, but they would be under pressure enough that there is tissue fluid leaking. And that would then build up in the lungs. How horrendous. And that could cause major problems with gas exchange because if your alveoli are sort of accumulating uh, 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 tissue fluid all around them, how will you have gaseous exchange, right? So proper drainage is really important, and that's why people who have these, disorders that arise from smoking or just generally, even without smoking, sometimes it's pollution or sometimes it's a genetic proclivity or COPDs, right? Chronic obstructive pulmonary disorders and stuff. These have a lot of these sort of fluid buildup problems because maybe the alveoli wall is not efficiently allowing gaseous exchange anymore, or maybe fluid accumulation is taking place because of an infection of some kind. And that could affect both gaseous exchange as well as lymphatic drainage, and fluid accumulation would then cause a major issue with proper oxygenation of the blood, enough oxygenation and enough um, and would not allow a person to take a full breath. So it would feel very incomplete and very uncomfortable. So best not to smoke is my solution here, because we don't really know, um, yeah, what are all these effects that occur from different environmental and lifestyle influences. Cool. Let's move to the next question. So, a direct relationship exists between interstitial fluid pressure and lymph flow. So, I mentioned what interstitial fluid pressure is, right? So, what clinical condition may arise if lymph flow is impaired despite rising pressure? So, can you guys think about what this might be? We talked about imbalances, right? So, what could the answer be here? Yeah, would others like to also give it a shot? I hope the question is clear, right? I'll explain it also. So the question here is, what would essentially happen if there is a problem with drainage? So either you have either you have inefficient drainage. You have low blood pressure, which leads to low amount of tissue accumulation. So uh, that could lead to different kinds of problems, or you have high blood pressure or normal blood pressure, but which leads to some X amount of tissue fluid accumulating, but it's not drained properly. So there's an imbalance in the lymph flow. So the if the lymph flow is not taking place properly, you can have accumulation of the fluid. So even if the pressure is rising and more fluid is accumulating, lymph flow is not sufficient to combat it, and so the fluid is building up. It very often happens in extremities like in the feet or in the hands, and that can lead to this condition, which is C option, edema. Okay, some people write it with an O-E-D-E-M-A, edema, right? And yeah, so that's seen in a lot of these cases. And in those cases, people may be asked to do certain exercises or take some certain medications. Yeah, or even in worst case scenarios, they might actually have to put in a little drainage. And I've read about that people have these sort of lymph drainage, sort of like artificial ducts that are sort of installed in their arms and legs to prevent this kind of drainage happening because of some, medical condition that's causing a major problem in lymph drainage, right? And so the other options, it's not the option, polycythemia. If you guys remember from, when you studied about blood and stuff, polycythemia is essentially an abnormally increased number of RBCs in the blood. So that doesn't really have anything to do here. Of course, polycythemia could have an effect on the blood pressure, I guess, which could then, have some repercussion on the lymph as well, but that's not really a direct, and that's not a cause, sorry, a result here. That's a cause. So The clinical condition that arises would not be the answer here. This would be maybe a clinical condition that may lead to, right? So it may be a cause, but not a result. And hypertension, this again is not because of it. It may lead to it. And hypoxia, again, it's just a decrease in oxygen content in the blood or an increase in CO2 content in the blood. Again, that's not really related here. Okay, so the answer here is edema. Okay, let's move to the next one. So I think three more and then we'll discuss some more topics. So in the intestinal villi, which you're familiar with, right? Which nutrients are primarily absorbed in the central lacteal rather than in the blood capillaries? So if you guys remember what the central lacteal is and what the characteristics of lymph are, then I think this one will be easy. I mentioned a certain characteristic um, molecule that you see a lot of in lymph fluid, more than you do in blood. So that would help you decipher this question. I can see some answers coming in. Yeah, so that's great. That's absolutely right. The answer is D. Yep, it's digested fats. So essentially, the lacteal is the lymph vessel that's found within the intestinal villus, and it's right next to the blood capillaries as well. That's also there in the villi, right? And the speciality of lymph fluid is that it takes up a lot of these fats. Um, so the digested fats are what enter into the lacteal. Water soluble vitamins, of course, they're not going to be the ones. So as you guys know, vitamins B and C are the water soluble ones, and the majority of them, which are A, D, E, and K, are the fat soluble vitamins. So maybe those you'll find more in your lymph in comparison to vitamin B and C. And monosaccharides and amino acids, these also are more hydrophilic and they enter into the blood directly or go via the tissues. But the central lacteal gets most of the digested fats. Cool. So why is it that fats transported via lacteals instead of directly going into the blood capillaries? So why is this particular process required? Right. Does anyone want to try? Yeah, great. Right, so the answer here is A option. So these fats or lipids are packaged into these structures called chylomicrons, um, which can't move through the blood capillary walls directly, so they actually need to go via the lacteals. Um, so fat absorption is like one of the like really major nutrient pathways, right? So alongside breakdown of protein into amino acids, breakdown of carbohydrates into your monosaccharides, um, and eventually glucose, you want you also have fat breakdown, right? But they don't directly go to the liver via the portal vein like they do in the other cases. It actually first enters the lymphatic system. So it happens inside your intestinal villus, right? Your intestinal villi, which has this lacteal. Um, and and the fats in this in turn are uh, sort of packaged into these chylomicron structures. So they are essentially lipoprotein particles that are necessary to transport fats. So why is it that they're in this way? Because fats, as you know, they're hydrophobic, whereas the liquid that they're in is aqueous medium, right? So they need to be packaged in such a way that they're able to move through an aqueous medium. So they are conjugated with lipid like apolipoproteins you may have heard of. So you have a lot of these fat breakdown products like triglycerides, you have some cholesterol, some phospholipids, and these are packaged alongside these proteins called apolipoproteins. Guys might have heard about ApoB, right? And so these are packaged all together. But what happens when that's done is that they're super big. They're really big, they're pretty hydrophobic, and they don't enter the blood capillaries. So instead, so instead they have to enter into the lacteal. Yeah. So I hope that's clear. Yeah. And why is it that they're only able to enter the lacteal, right? So it's just because blood capillaries have smaller gaps, whereas lacteals have larger gaps, and no sort of basement membrane that's like it has these tight junctions and stuff. So it's more permeable to these kind of larger particles like chylomicrons. So they then go into the lymphatic circulation, and yeah, so they are eventually broken down and deposited. So that's part of the digestion part. So I won't go into that right now. Yeah, cool. So I hope that's clear. Now, the last question is, of course, a dysregulation related question. So, elephantiasis, as you guys studied, results from which organism that obstructs lymphatic vessels over time? Yeah, great. So, I think you guys have answered mostly. Yeah, so the correct answer, as you guys have said, is D option, which is Buscheraria bancrofti. So that's quite a mouthful, but basically, elephantiasis is this obstructive disease of the lymphatic system, and it's caused by these gross parasitic worms, which is this Wuchereria stuff, and they are transmitted via mosquitoes. So this is what causes filariasis essentially of lymphatic filariasis, right? And what happens is that the mosquito, when there's like two phases of its life cycle, right? One inside the mosquito and one in the human, which it infects. So the mosquito, when it bites a person, it injects the larvae this uh, of this parasite into the body, and these migrate to and via the lymphatic vessels. And then they sort of mature into worms inside the lymphatic, and they can live for like a while. Okay, and they're not immediately detected unless you're really lucky, I guess. And these adult worms can then excuse me, they can cause inflammation, they can cause fibrosis, right? They can cause blockage of lymphatics, which is one of the major sort of um, red flags here because that's what leads to a lot of accumulation, as we mentioned, and a lot of lymphedema, which is essentially edema caused by lymphatic um, accumulation. And so the tissue sort of gets really thick, and then patients who have this get a lot of highly accumulated, highly swollen legs. It swell in the scrotum, in the arms, in the breasts, and all of these regions it may become super severe where someone would need to then have it artificially drained, or it can even go worse, and it can lead to dysregulation in the entire in the immune system because you'd have effects on lymph nodes and all sorts of chronic problems. So there are treatments, though. I believe I think ivermectin can be used, and also people do have like surgery for, cleaning it up and draining the lymph and so on. Yeah. So that's about the lymphatic filariasis or elephantiasis. Okay. So that's one set of questions. So that was about the lymphatic system, lymph nodes, and lymphatic drainage, right? So if anyone has any questions, please ask whenever you need to. With that, we can move to this next part about the excretory system. So this part, I'm sure most of you are very clear about. I'm sorry, which is the components of the excretory system. I'm not going to go into too much detail with them, but essentially, you have most people, though there are people who just are genetically sort of born with only one kidney and they're fine. Um, Most people have two kidneys. You have a renal artery carrying blood to your kidneys, a renal vein that takes deoxygenated blood away from the kidneys. You have a ureter that leaves the kidney with formed urine and deposits it into the urinary bladder, which has very interesting contractile tissue in it that allows stretch sensing to then it like talks to your brain essentially and tells you when you need to pee. And then a urethra through where the which has a sphincter that is relaxed when you are urinating. So, and you have these little caps on the kidneys called adrenal glands, which are very, very important centers and endocrine centers for hormonal regulation, which will be discussed later. So, those are the macro components of the excretory system. And if you sort of take a cross section the of the kidney, you will see that there is a renal medulla, and you have the renal cortex where you have these nephrons beginning, and these are the renal columns in between, and this is the ureter here. So you have these collecting tubes that sort of all come together and empty out formed urine into the ureter, right? Okay. So you guys read in your coursework about glomerular filtration, right? And how there are these different this functional unit within the nephron is the functional unit of the kidney, and you have blood that is filtered at the level of the nephron inside the arteries of the glomerulus, and then that filtration is what allows osmotic balance to be maintained in the body, right? So, yeah, so some basic questions regarding that I think we can go through, and then we can go into more detail in the upcoming slides. So, I'll sort of explain things as we go. So, do send the answer. Which of the following one is considered only one of the several major functions of the kidney? So, essentially, which of these is a major function of the kidney amongst the others that are not? Yeah, cool. Yeah, perfect. So that's exactly right. The removal of nitrogenous waste is one of the major functions of the kidney. I think I have other questions on like what the nitrogenous waste is, so I won't talk about it right away. Of course, the kidney doesn't produce digestive enzymes, it doesn't really regulate body temperature, and it does not at all store glucose. That would be catastrophic. So, right, the answer here is A option. Okay, so now if water intake were to decrease while body water loss increases, the kidneys respond by doing which of these following activities or which of these mechanisms? So this is like a homeostasis question, right? How is the kidney maintaining homeostasis in a condition where there are external environmental cues that may cause some tension in the body or dysregulation in the body? So imagine if you are outside in the sun and you're walking around and it's really hot and you're sweating, but you forgot to bring your water bottle. So you're not drinking water, but you're losing water actively in the form of sweat. So what would the kidneys try to do to make sure that I mean, it can only do up till some certain degree, after which things will start going wrong. But what would it do to try to maintain homeostasis? Yeah, exactly. It's option C, which is reduce the urine output to conserve water. It can only do it so much, right? It cannot. So the thing is, why do we need to urinate, right? Why is there a need for excretion? Because you're removing a toxic buildup of nitrogenous waste in the body. You want to make sure that you don't have these wastes building up. And if they do, then that can cause some real major health issues. So there is only so much that we can sort of stop and conserve, because if you do that for too long, you could actually build up a lot of these wastes. So increasing urine output doesn't make sense. You just lose more water. Maintaining the same urine volume, again, that would only be if you were able to drink water again, which if you're not doing, then that would be a problem. If you eliminate more sodium only. So in my last lecture, I had mentioned how the trick to remembering sort of the absorption of salts and water and how that works in the kidney is to remember that where sodium goes, water goes, or where salt goes, water goes. So if you're eliminating sodium, along with that, you will eliminate water. So if you're eliminating more sodium, you are doing the opposite of what your body needs in such a stressful condition. You're actually losing water. So the only right option here is C. Great. So I hope that was clear for everyone. Let's move to the next one. So the kidneys help maintain acid-base balance primarily because amino acid breakdown releases which of these components? Right. Does anyone want to try? Yeah, so the correct answer here is like it is hydrogen ions. So there is hydrogen ion release upon amino acid breakdown that directly helps with acid-base balance. So essentially, how it works is that when the amino acids are metabolized, especially, there are these amino acids that contain sulfur, right? Like methionine and cysteine. So they generate these non-volatile acids, which then release H plus ions. And so these H plus ions are secreted by the kidneys to maintain blood pH. So it's not just amino acid breakdown, but there's like an acid load, and that's what the kidney uses to restore the balance. So essentially, these sulfur-containing amino acids, they form like sulfur-containing acids, like sulfuric acid and so on. And there are some other, phosphoproteins and all, and you have these different types of organic acids and so on that are created, and then those lead to release of H plus ions. And yeah, and that's what regulates the pH. So if you have more, then it will lower your pH. And so then, you could have, if you have too much of this, it can lead to something called metabolic acidosis. So there's a constant acid production. So for people who eat protein daily, like I don't think it has to be meat, but I would assume it's probably easier if you're eating meat, that you would have acid production that is continuous, because you would have continuous protein to break down into amino acids, which you would then break down into um, these different types of acids that release H plus, right? And bicarbonates actually are reabsorbed. So it's filtered at the glomerulus, and the kidneys actually reclaim almost all of the filtered bicarbonate and prevent its loss in urine. Why? Because bicarbonate does the opposite, right? It buffers the H plus, but they are not produced from amino acid breakdown. So bicarbonate and hydrogen ions are both really important for maintaining the acid-base balance. But the one that's produced from the breakdown is of amino acids as H plus. Okay, cool. Um, right, let's move on to the next question. So I had mentioned about the nitrogenous waste. So uric acid in humans is mainly produced from the breakdown of which of these products? Yeah. Do others want to try? Right. Yeah, so the correct answer here, yeah, you're absolutely right. It's D option, which is purines such as DNA and RNA in the cell. Um, as you know, DNA and RNA contain nitrogenous bases, so obviously you would have nitrogenous wastes that are generated from the breakdown of these components, and uric acid in humans is mainly produced from their breakdown. So uric acid is one of the nitrogenous waste types that humans secrete alongside urea. And I'll come to an interesting point. Maybe I'll finish these questions, or actually, I can tell you now. So there is an interesting topic with respect to sort of an evolutionary um, determination of what type of nitrogenous waste is secreted by which type of animals. So, based on water availability, you could have differences in which kind of nitrogenous waste that particular organism excretes, sorry. So, if there is toxicity of so the you have to balance toxicity of that nitrogenous waste along with water and energy availability, and that obviously is tied in with the habitat. So what does this mean, right? So you can have different types of nitrogenous waste. You can have ammonia, and you can have uric acid, and you can have urea. So which ones are we, right? We primarily excrete urea, right? Birds do uric acid, and ammonia is typically for um, like fish and water dwelling creatures. So animals that excrete ammonia, they're called ammonotelic animals. I'm sure you've heard of this, right? And these are the ones who excrete ammonia. So they're like aquatic invertebrates. And why is it that aquatic invertebrates, which are these, water dwelling uh, organisms, why is it that they are ammonotelic? It's because ammonia itself is really toxic and it's really soluble, and that's why it needs a lot of water to excrete from the body. Okay, so you don't need too much energy to do this, you need a lot of water. Now, suppose someone like you and me who lives on land, or a bird that's typically just flying around, they don't have a lot of water available at so easily to be able to excrete something like ammonia. So if ammonia were to build up in our bodies, it would be really toxic for us. So that's why aquatic animals use the ammonotelic um, form, the like method of nitrogenous waste excretion, because water is super abundant and they don't really need to convert it to anything. And so they just directly leaves their body through their gills or other body surfaces. So they don't need much energy to do it, but you need a lot of water. Then there's ureotelic animals, who are the ones who secrete in the form of urea. So mammals like us and adult amphibians, like frogs or toads, they excrete urea. So urea is relatively less toxic than ammonia. It's also water soluble, and you need like a moderate amount of energy, because you have to actually undergo this urea cycle, which you may have heard of in the liver, and it needs like a moderate level of water for excretion. So we're sort of in between, right? In terrestrial lives, like terrestrial life, like mammals, we have some level of water conservation that we need to do, and for that we need to have reduced toxicity of that nitrogenous waste. So this is a good sort of in between compromise point, right? So it's safer than ammonia in terms of toxicity. It's not as water demanding. It's not too energy expensive. So we're right in the Goldilocks zone per se. Now you come to euricotelic animals, right? So these are like reptiles, insects, birds, okay? These excrete uric acid. So this is the least toxic of all of the nitrogenous waste, but it's really poorly soluble in water, and it's excreted not as sort of liquid um, excreta, but it's actually more like a paste, like, or crystals. And it requires a lot of energy to do this, but it's because the birds and snakes and reptiles, they really need to conserve water, because they're not in typically in conditions where they can have access to that much water and have water-based excretion systems. So uric acid essentially just precipitates, and then that is excreted out, but it requires a lot of energy to do the breakdown products. Yeah. So this is just some interesting sort of evolutionary adaptation that I thought I'd talk to you about, um, which explains why our primary mode of excretion is of nitrogenous waste is via the form of urea, and we are ureotelic organisms. Okay, now after that aside, let's go to the next question. So when sodium intake suddenly increases tenfold, what is the most immediate renal response? So this is like a hypothetical. So I just mentioned the relationship between sodium and water. So applying that over here, which do you think the answer is? Okay, so let me explain this one. If anyone else wants to try answering, please go ahead. Yeah. Oops. Just a sec. Yeah. Cool. So, the right answer here is increased sodium excretion in urine, which is the C option. So, why is this the answer, right? So, why is it not A, which is suppression of glomerular filtration? So, if you suppress it, what would happen, right? If you're suppressing it, less would filter through. So you just have a retention of all of this crazy amount of sodium. And remember, where sodium goes, water goes. So if you have retention of all of the sodium, you have retention of all of that water. So you suddenly are retaining an excessive amount of water in the body, which can lead to disruption in osmotic balance, disruption in blood pressure, all sorts of problems. Increased potassium retention, that just doesn't make any sense. And complete cessation of water reabsorption, that also would not um, solve the problem here, because if you stop reabsorbing water, your sodium level is still really high. So even if you're not able to remove the sodium, then that could cause any kind of major sort of physiological problems. So, the best thing for the kidneys to do in that point is to increase the sodium excretion, which is possible by just reabsorbing less sodium, right? In the PCT and the DCT, it can, and the loop of Henle, it can essentially regulate how much sodium is reabsorbed. And if it reabsorbs less, then more of it is excreted into the urine. Along with that will go some water. Cool. All right. So next question. Yeah. So what does extracellular fluid volume, why does extracellular fluid volume rise when sodium intake is very high? So again, this is just following the simple concept that I talked about. So applying that, let me know what you think the answer is. What do you guys think? Yep. So everyone's got it right. Good job. Yeah, great. So it is B option. Increased osmolarity promotes water retention. So like I mentioned, where sodium goes, water goes. So if you have If you suddenly eat a whole bunch of salt, then and now you have really high sodium in your fluid, then that also to sort of essentially it's like to dilute that, to counteract that, you will have a rise in extracellular fluid volume. Okay, so that increased osmolarity essentially of the extracellular fluid because of increased sodium content promotes water retention, and that water essentially dilutes out your sodium in the extracellular fluid, right? So yeah, that's the correct answer here. Okay, so which structure carries filtered urine from the kidney to the urinary bladder? This is just an anatomical question. I hope you guys know it. Yeah, great. I think anyway, like this one is pretty clear. So it is D option, which is the ureter. So each kidney has a ureter that arises from the collection of those collecting ducts that are like the end point of each nephron. So all the nephrons have these collecting ducts where the finalized urine is reaches, and then all of those sort of come together and they form this macrostructure, which is the ureter, which then extends downwards into the urinary bladder. Okay, so that's that. Let's do one more. So, what percentage? So, this is just another anatomical question. So, what percentage of nephrons are cortical nephrons? So, you have to remember that the kidney has a division, right? So, it has this medullary region as well as the cortical region. So, um, yeah, yeah. So the answer, yeah, you're right. The answer is C, which is around 80 to 85%. So just to sort of jog your memory in case you've forgotten. So in the kidney, there are, so the functional unit is nephrons, right? And there are two major types of nephrons. So you can have cortical nephrons and you have these medullary or juxtamedullary nephrons. And the difference between these nephrons is either, of course, their location. Then they have different loop of Henle length, and their blood supply and the role that they play in final production of urine is different. So the cortical nephrons, they are located in the outer cortex of the kidney. So you remember the kidney shape, and so the outer peripheral region, that's where they are located. And their loop of Henle, right? So you guys remember loop of Henle. So you have the proximal convoluted tubule, it dips into the loop of Henle, and then you have the distal convoluted tubule ending in the collecting duct. So this loop of Henle dips only slightly into the outer medulla. Okay. And this is the majority of nephrons, so 80 to 85%. And yeah, they don't have a very extensive blood capillary system. So the vasa recta, as you guys probably remember from school, so you have that capillary system, so it's not very extensive. And their major function is filtration and bulk reabsorption and forming the basic urine. Okay, so they're not very specialized for concentrating the urine, and that's the majority of them. Now, these special guys, which are the juxtamedullary or medullary nephrons, they are located, um, they have their glomerulus near the junction between the cortex and the medulla region. Okay. And they have a very long loop of Henle, which goes all the way into the inner medulla. So the cortical nephrons have a loop of Henle that just about touches the medulla, whereas these guys, the medullary ones, have a loop of Henle extending deep into the inner part of the medulla. And they are the, relatively smaller amount of them are there, so maybe 20 to 15 to 20%. And they have this extensive vasa recta blood supply and a lot of these capillaries that are sort of looping in. And these are the guys that create medullary osmotic gradient and enable the concentration of urine. So they're very, very important for water conservation. So why is it that this loop is important? Why do they have such a difference in their loop length? So the descending loop of the loop of Henle, so if the loop of Henle is, imagine this kind of a U shape, right? So the descending loop, the one that's coming down here on the left, that is permeable to water, while the ascending loop going up in this way, that is impermeable to water but pumps a whole bunch of sodium chloride out. And this is what creates this, what is called a countercurrent multiplication system. Essentially, it causes the medulla to become hyperosmotic. So that has a high concentration of salt, and it allows some hormonal input that allows the concentration of urine to take place. So the cortical nephrons on their own can't do that, which is why you need the medulla to cause this hyperosmosis, osmotic tension, which then allows this. So yeah, I hope that was sort of clear to you. Yeah. Okay. Now here's a slightly thinking question. So if a person suddenly decides to make the weird decision to drink 3 liters of water in one sitting, what steps would the kidneys need to follow to establish osmotic homeostasis again? So imagine there is some person who decided to drink 3 liters all at once. So what would happen to them? Does anyone want to try? So there's no there's no options here, but if someone wants to say it or write what they think into the chat, that would be cool. Three liters is quite a lot, I think, right? I don't think I could drink 3 liters in one go. Hmm. Right. So if someone were to suddenly do this, their body would temporarily become like hypoosmotic, right? So that means their plasma osmolarity would fall way below, or at least to some degree below what is normal, which is around 300 milliosmoles, right? So if their plasma osmolarity falls, the kidneys need to act ASAP to restore this osmotic balance, right? So they have to do a bunch of things to make sure that this osmotic homeostasis is maintained. So, of course, you have receptors in like the hypothalamus that can sense that the blood in the body has got diluted because this crazy person has drank so much water. And so this will suppress the release of any hormones, um, like antidiuretic hormone from the posterior pituitary. So if you guys remember, the hypothalamus talks to the pituitary, the back part or the posterior part of the pituitary releases this hormone called ADH, antidiuretic hormone, also called vasopressin. So antidiuretic hormone is basically and it's basically stops you from peeing. So if we suppress it, that means that the body should then be able to urine, like the person should be able to urinate as much as possible. So now when you inhibit it, then the body essentially shifts into a mode to eliminate as much water as possible. Now, another thing, so that's at the level of hormonal regulation at the level of the brain. and sensors of osmolarity of the blood, right? And now the kidney also needs to do something, right? The kidney would need to then make sure that it's not reabsorbing as much water. So typically there's some X percentage of water that it's reabsorbing at its different parts, but it needs to not do that. Instead, it should, and this of course ADH helps with that. Like if you have low ADH, then the collecting ducts and stuff, they become a lot less permeable to water. Um, and so you have a lot more water that just stays inside these tubules of the nephron, and that allows a lot more water to be released. And so then that allows the kidneys to then generate a lot more of this diluted urine that then allows the excretion and balancing out. So now you have to be careful here as well. If you are releasing so much water, you want to make sure that you don't release all the sodium that you have, right? So you don't want to remove sodium levels and disturb the sodium levels, but you want to remove excess water. So you have to make sure that the solute loss is minimized, but the water itself is excreted. So in such a case, you want to, I'm sure the loop of Henle is super important because you have differential permeability in that case, right? So then finally, with all of these different steps, you'll be able to come back to blood osmolarity close to about 300 milliosmoles. And then eventually things will go back to baseline and ADH will be secreted normally again. Yeah. So cool. That was just a sort of thinking question. Yeah, and this is just to recap what we saw so far, which is the structure of the nephrons. So you have the kidneys macrostructure here with your the capsule that surrounds this whole kidney. You have the renal pyramids, renal columns. This is the cortex. Inside these pyramids is the medulla. This region is called the calyx, which then extends through this way into the ureter. This is the renal artery, the renal vein. Yeah, and this we discussed, and this is the nephron. So you can see over here, this is the afferent arteriole, and we discussed how it's important. In last class, we discussed, right, that it's important that the afferent arteriole has a larger diameter, a cross-section diameter, right? Because, in comparison to the efferent arteriole and in comparison to the capillaries in the glomerulus, because because you want to maintain high pressure in these capillaries in comparison, and that would be allowed if you have a higher dia over here. So more blood will then be filtered into a smaller volume. So that's more filtering that's allowed. And then you have your glomerulus encased in the Bowman's capsule, and you have your proximal convoluted tubule entering here, and then you have your distal convoluted tubule over here, which is then extending into your loop of Henle, and these are your peritubular capillaries that surround the loop of Henle. And then finally you have this leading into the collecting tube, which then goes into the ureter. So that's just to um, summarize, right? And it uh, they're the functional unit, and you have about 1.25 million nephrons per kidney. Yeah, so they're literally the bulk of the kidney, and then you have other supporting tissue that would do some other supporting functions, but this is the main thing, right? Cool. So I had some questions here that might require some thinking. If you guys want to try it. So if the total renal blood flow is say 1100 ml or 1.1 liters per minute, and plasma is about 55% of the blood, right? Then what is the approximate glomerular filtration fraction or the percentage of plasma that is filtered? So I know that some of you are maybe like on the move or not necessarily sort of able to get a pen and paper immediately to try this out. So I'll just explain it anyway since we are running out of time. Right, so the options here, the options here are like 10%, 20%, 40%, 55%. Right, so how do we go about looking at this question? So essentially, per minute, you have about this quantity, which is 1.1 liters of blood that reaches the kidney every minute. Roughly 55% of the blood is plasma. So 55% of 600 of 1100 ml is roughly 600 ml. Okay, roughly. Now this needs some prior knowledge, which is what is the glomerular filtration rate or GFR. So you guys learned about GFR, right? Essentially, how much is filtered per minute in the glomerulus. So the GFR in the normal person's kidney is about 120 milliliters, ml. So the filtration fraction would then be what, right? If you have roughly 600 ml of plasma, right, which is 55% of 1100, if you have about 600 ml of plasma reaching the kidneys, and every minute you have 120 mils, that is actually the filtration that's filtered by the glomerulus, then your fraction of filtration or the percentage of plasma that's filtered is 120 divided by 600 into 100 to convert it to a percentage, right? So if you do 120 divided by 600, that's 0.2 into 100, that's 20%. So the answer here is B option, which is 20%. So this is just basically testing whether you remember what the GFR of a normal kidney is, which is 120 ml. Okay. So if you know that, then the rest of the question sort of falls into place. So the answer here is B, 20%. So this is not clear, you can let me know. I'll also be posting this this PPT with the answers. So if you are confused, you can always refer to it later. Okay, so I had one more question. Actually, I have a bunch more, but I don't think we'll be able to finish everything in this session. So we can always look at it in the next session. So let's just maybe complete a few more and then we can get done for the day. So a patient, right? This is just an example. If a patient develops severe hypotension, which can cause glomerular capillary pressure to fall from say 60 mmHg to 50 mmHg. Now assume that the oncotic pressure, and we talked about what oncotic pressure and capsular hydrostatic pressure are, right? So assume that these are the values and these remain unchanged. What will most likely happen to the filtration in the kidney? So this, I think, requires you to just remember what is net filtration pressure, right? So net filtration pressure, so there's definitely something happening in this person, right? What do they mean? Like they have severe hypotension. That just means you have very low blood pressure, like really low blood pressure, right? And so that could affect the net filtration pressure at the level of their kidney also. So the net filtration pressure is, the equation works out to glomerular pressure, right? So glomerular pressure, which is where is it? Yeah, here, this is your glomerular pressure. And that minus the oncotic pressure, which is this, and the capsular pressure, which is your hydrostatic pressure. So normally, then how would you calculate your net filtration pressure, you would have glomerular pressure, which is say 60, which is your normal value. So 60 minus 32, which is your oncotic pressure. And so 60 minus 32, right? So that's like 28. And then further minus your capsular hydrostatic pressure, right? So you're essentially removing the two opposing pressures, you're minusing them or they're canceling out. So 28 minus 18, which is 10. So roughly 10 mmHg normally would be the net filtration pressure. But if this person's has hypotension and their capillary pressure, glomerular capillary pressure falls to 50 mmHg, what would happen? Right? Then it would be 50 minus 32 minus 18, which is 0. Okay, so it's roughly 0 would be their net filtration pressure. So what happens then? There's no driving force left for filtration, and so urine production would just not happen. And that would explain why, if you have a really dangerously low blood pressure, that can rapidly lead to kidney failure. Okay, so everything is linked up, right, to blood pressure. So very low blood pressure, severe hypotension that's sort of sustaining for a while and it's not, yeah, ameliorated with some medicines or something, that can lead to rapid kidney failure because the urine production will just stagnate because net filtration pressure has become 0. Okay, let's do one last question, I think, for today, and then we can adjourn. Because I have more, but I think we won't have time. So, okay, imagine that there's a kidney stone obstructing the ureter while ultrafiltration is continuing. So, which mechanism amongst these would best explain the eventual decline in GFR? So, we talked about GFR, right? It's the glomerular filtration rate. So, does anyone know which one it might be? So, you can think what is happening if you're obstructing the ureter with a kidney stone, right? Urine is not able to move properly from the kidney to the bladder or basically into the reservoir, right? So you have urine accumulating in the ureter, or it's moving very, very slowly because maybe the opening has become very, very limited. So that would lead to a decline in GFR, but how, right? Yeah, great. Good job. So the guys who answered B, you're absolutely right. That's the correct answer, which is you would have increased capsular hydrostatic pressure that opposes filtration. So how does this, so why is that the answer? So when the urine can't drain out because of this obstruction, fluid is essentially accumulating in the kidney, in the Bowman's capsule. And this raises the capsular hydrostatic pressure. Makes sense? And this directly opposes filtration. So as this pressure approaches glomerular pressure, the net filtration pressure will become close to 0, right? And that would again cause a problem with filtration. So again, what's the deal with pressure, right? If it's what drives a gradient. You have to have higher pressure on one side and lower pressure on the other that drives the gradient that allows fluid to be pushed from one side of, say, the membrane to the other side. But on both sides, if it's the net filtration pressure is becoming 0, you potentially are just halting this filtration altogether. And that makes this urinary obstruction a super big medical emergency, right? Because yeah, and so that's what. So you have your net filtration pressure again approaching 0. That's why this leads to this eventual decline in GFR. Cool. Great. Oh, yeah. So, mhm. So does anyone have any questions? Any other questions? I have a few more questions that we can go through if you guys want to. Um, so this that you can see here, this is a renal corpuscle, right? So and next to it you have this juxtaglomerular apparatus. So all right, so I can just explain it in some varying details. So essentially, the renal corpuscle is where blood is filtered, and the juxtaglomerular apparatus is where blood pressure and filtration are regulated. Okay, so so that's all of this. Okay, so the renal corpuscle has two parts, right? It has the glomerulus, and it has the Bowman's capsule, which is around it, right? And that's the initial filtering unit of the nephron. So the glomerular capillaries, they are like this knot of capillaries that they're really porous and they operate at obviously a very high pressure, which is, as we talked about earlier, it's roughly 60 mmHg. And these allow water and some small solutes through, and they retain cells and large proteins, obviously. And this is not like just an exchange capillary bed, it's designed for filtration. So there's not necessarily any nutrient exchange over here. Okay. So the glomerular capillaries lie between two arterioles, the afferent and the efferent. Yeah, so the afferent arteriole brings blood into the glomerulus and efferent is taking it away. And constriction of the afferent arteriole, which you can sort of see has a higher diameter, though it's not very well shown here. Constriction of it will lead to reduced GFR, whereas dilation of it basically means that this would dilate, there'd be more blood, that would lead to a higher glomerular filtration rate. Okay, and meanwhile, the efferent arteriole, this guy, that carries blood away and it doesn't become a vein immediately. It just goes into the peritubular capillaries. It's not shown here, and it goes into the vasa recta, which goes and surrounds your juxtamedullary nephrons, right? Those 15 to 20% of nephrons that we talked about, right? And they are narrower. So this is narrower and this helps sustain filtration pressure. So it's sort of like blocking a hose pipe, right? So the pressure builds up upstream and allows it to be filtered at a certain pressure. And then you have the Bowman's capsule, right, which we talked about. It's a double-walled cup that catches this filtrate from the glomerulus. And it has two layers. It has the parietal layer and it has the visceral layer. So the parietal layer, it's the outer wall. This is the parietal layer you can see over here in this color of blue. And it has simple squamous epithelium and it doesn't really do much filtration. It's mostly structural. And then you have the visceral layer. Okay, and this is the real filtration partner because it has these cells called podocytes and pedicels. As you can see, that's drawn here. So these are weirdly shaped dudes are podocyte and these smaller ones are pedicels, forming the visceral layer. And between these cells, between the little triangles, you have some spaces, some slits that are the final barrier before the filtrate from the glomerulus enters into the corpus, into the capsule. Okay, so yeah. Mm, cool. And if there's damage, you guys might have heard about this condition. If there's damage to the Bowman's capsule and this filtration, you might have too much stuff being let in. So you actually have protein in your urine. So you should not have protein in your urine. But if there is damage in the Bowman's capsule region, that could lead to excessive protein just entering into the capsule, even though it's not supposed to. That can cause a condition called proteinuria. Okay. So yeah, that is that. And then yeah, there's some other things. Um, yeah, and one of the main important things here is this juxtaglomerular apparatus, right? The JGA. That is like the pressure sensing device of the kidney. It's literally very important for deciding whether it's going to increase blood pressure, increase filtration. Remember that there's like renin secreting cells there and stuff. Yeah, and this macula densa, there's these specialized cells in the DCT that touch the afferent arteriole, and they are important for measuring sodium chloride concentration in the filtrate. So that helps you sort of make a decision if there's low NaCl, low BP, low GFR, high NaCl, high filtration, so on and so forth. Yeah, so that's the main thing. And as I mentioned, the juxtaglomerular cells, these have some granular cells that secrete renin, which is very important for the RAS or the renin-angiotensin aldosterone system that helps increase blood pressure. Okay, so if there's low blood pressure, that triggers renin release, and that will then cause the blood pressure to increase. Yeah, so those are the major things. And as we discussed earlier, you have the PCT, proximal convoluted tubule. All of it is not shown here, but this is where it starts. And here is where a humongous amount of water reabsorption happens, right? So we talked in earlier classes about how much water goes in the proximal tubule. So that's about 70% of the water is reabsorbed at the PCT, along with sodium, some glucose, some amino acids, bicarbonate, and so on. And it has this you can see these little things like a brush. So this is a brush border microvilli, and these increase the surface area by a lot, and it improves absorption. After that, you have the DCT, the distal convoluted tubule, which isn't shown here, and that is sort of close. Its main function is to fine tune the electrolytes, and aldosterone, right? You remember from the RAS system, that's the one that comes and acts over here. So it's in charge of like 5% of the sodium reabsorption. So it's what acts over there and helps sodium reabsorption to increase so that blood pressure can increase. Yeah, so it's sort of like adjusting precisely. And yeah, that's pretty much it. So that's this system. And then I had more questions, but there's no time. So I'll just upload these and you guys can take a look, or we can discuss them in the next class, which probably makes more sense. So we can discuss these questions in the next class. There were just a couple of them, but it's better to discuss and understand why something is a certain, why the answer is a certain thing. So I'll continue from here in the next class and then cover more topics. I hope that's fine with everyone. Yeah. So that's it for today's class then. Um, Hope you guys found it useful. And if you have any questions, you can always ask. And I'll see you guys in the next class, next Tuesday. Okay. All right.